Heard about PCR biosystems?
There's quite a buzz about us. Hop on over to our website to find out more.
Welcome to Pint of Science 2021. Cheers to everyone. Hope everyone's having a great evening so far. And it is day 2 already. Amazing. We had some fantastic talks to start things off yesterday. And I'm very excited to be your host today. My name is George Finney, and I'm hosting from the lovely city of Glasgow. And the University of Glasgow is hosting this Our Body Pint of Science night called Hijacking the Immune System for Better or for Worse. I'll give you a quick rundown for our session tonight. You're going to have 2 wonderful talks with some very good friends of mine who both work in the chemokine research group at the University of Glasgow. Firstly, we'll have Dr. Alan Hayes, who's going to talk to you about how diseases such as cancer have very creative ways to disrupt how immune cells travel around the body. And then after that, you'll hear Dr. Marieke Pingen, who'll show that even though mosquito bites and the viruses they transmit are pretty pesky, but diving into how skin deals with them can help us learn a lot more about how to tackle how they spread. 2 things I'd like to note for everyone is in between Alan and Marieke's talk, so during Alan's talk, please feel free to ask questions. But what we're going to do in the interim is I want to hear from you guys about any fun immunology facts that you think are really worth looking at. And we'll go through those in the intermission between both talks. During Alan and Marieke's talk, please feel free to put into the comment section any questions you would like us to ask, because at the very end of the show tonight, we'll have a live Q&A session with both Alan and Marieke going through all the questions that you want to go through. And without further ado, I will hand you over to our first speaker, Alan, so we can talk about how immune cells find their way to fight disease.
Hi everyone. As you just heard, my name is Alan and I'm from the Chemokine Research Group at the University of Glasgow. And tonight, I'm going to give you a quick talk about how we can help immune cells find their way through the body. So, how do our bodies actually stay healthy? Well, the human body is actually designed quite like a fortress. So if you think of a fortress, it's got lots of external walls and barriers that are designed to help keep any invaders out. The human body is designed in much the same way, and inside we have all our internal organs that we want to keep nice and healthy. And the skin acts as this external barrier to help prevent invaders from getting inside. But if you think of a fortress, walls alone can't stop invaders. So just like the walls of a fortress can be breached, our skin can get damaged, and this creates points of weakness. So in a fortress, you might blow up the walls with some dynamite, and this would then let you get inside. Our skin, we can get puncture wounds, or we could fall and graze, say, our knees, and this creates entry points that pathogens can exploit to get inside our bodies. In addition to these, if you think of a fortress, it's designed to protect something inside, so there has to be a way to get in and out. And much like that, our bodies also have to have natural entry points, such as our mouth and our nose, to allow important bodily functions to take place. So on a fortress, your walls will be protected by archers. So up here on the ramparts, you have a couple of archers who can shoot any invaders who might try to get in the fortress. You're also going to have several warriors that will patrol the inside of the fortress to kill anything that makes it inside. So how does your body achieve this same effect? That is the immune system. So the immune system acts as the foot soldiers to mimic what the warriors would do inside a fortress. So if you look on the left here, you can see the immune system is split into 2 main parts: the innate immune system and the adaptive immune system. You don't need to worry too much about what these different parts of the immune system do. But what you need to know is that all the cells you can see in that diagram all work together to help kill any invaders that make it past the barrier that our skin provides. But what is the immune system exactly? So it's a bunch of cells that are there to deal with any threats that make it inside our bodies. And our bodies are constantly under attack by various different types of pathogens. These can range from bacteria, parasites to fungus and viruses. All 4 sources of infection can be relatively mild, such as you might get a virus like the common cold that's not overly deadly, but then you might get a virus like Ebola, which is much more deadly, and all four sources of infection can range between mild to very severe. But luckily, our immune system is a natural defense against these pathogens, and it's very good at first identifying threats. All those cells can then work together to kill the threats, and then we can generate immunity. When we generate immunity, that is when we get long-lived cells that can make antibodies, or they remember what they've just killed, so that if we meet the same infectious agent a second time, our body can respond to it very rapidly and kill it before we actually get sick. We can mimic this with vaccines, and that's why vaccines are super important at stopping the spread of pathogens, because it mimics as if you've already seen it. So that's why vaccines like the COVID-19 vaccine are really important for helping keep a population healthy. But our immune system isn't only designed to deal with invaders. It's also got a role in helping protect us from cancer. So cancers will arise when some of our cells mutate. So when cells divide, the DNA is replicated and split across 2 cells. When this happens, sometimes errors can occur. However, when those errors occur, our immune system is very good at killing those cells. However, sometimes those cells do escape detection, and they can then grow into a tumor and become a cancer. So how does the immune system work in general? So in order to get an effective immune response, they generally happen in what are known as secondary lymphoid tissues. And these are the spleen and your lymph nodes. So your spleen is located in your lower abdomen, while lymph nodes are located throughout your entire body. You can find them up in your neck, right down to your knees. And they're connected by a dense network of lymphatic vessels that allow cells to move from your various tissues to your lymph nodes and between lymph nodes. So this allows your cells to easily move around your body, to talk to one another, to easily eliminate threats that might make it inside. But how do your cells know where to go? Because our bodies are pretty complex. So if you look at this Lego figure on the right here, inside there's lots of internal organs. You've got the guts, liver, stomach, brain, heart, lungs, and any one of our tissues can be infected at any time. So our body has to have a system that allows cells to get to where they need to go at the right time, and that's where chemokines come in. Now, you may never heard of chemokines before, but I'm just going to break them down quickly and let you know what they do. So they're small molecules that our body can produce, and they act as a chemical trail throughout the body. This chemical trail can lead cells around the body to help your immune system get to the right spot to deal with any trouble. But how can our cells sense this, and how can they follow it? And they use something called chemokine receptors, which allow them to follow specific chemical trails. So if you look at the diagram here, you can see the brain has a plus symbol, the lungs have a circle, and the skin has a diamond. These are what chemokines kind of function like, and that each tissue has its own specific combination of chemokines that let our cells know where they are and how to get there. Almost like an address code. So if you were sending a letter to someone, you would use a postcode to make sure it gets to the right spot, and chemokines do that same thing for our bodies. And this is what chemokines look like. There are lots of chemokines, and there are lots of receptors. And this diagram is a bit of a mess, with arrows going everywhere, with chemokines interacting with receptors, and receptors acting with multiple chemokines. So you'll be forgiven for feeling a bit like this. And even someone like me who works on these sees this diagram and feels the exact same way. But believe it or not, sometimes our cells can also get really confused by the complexity of this system. So just like if you were reading a map, you might get lost and take a wrong turn. Sometimes our cells can't quite read the address codes that our body has laid out for them. So if we look at this in the context of cancer, you want cells to get in there to eliminate the tumor. So you can see here, there are 2 cells. You have your pinky purpley cell and your yellow cell. If you think of this like a party and you're trying to get to your party, the purple dots you see are the chemokines which act as the address code. So if you're the yellow cell, you don't know what the address is and you can't get to the party. However, if you are this pinky purple cell, you can easily move into the party, do your job, and eliminate any threats that you find. What happens if we take away those purple cells that can respond to these chemokines? We are left with a yellow cell that can't move into the tumour. And what's going to happen? It's going to wander the other way, and this then lets your tumour grow and spread, which obviously we don't want, as this is going to be very detrimental to our health. So that's the first way cells can get lost. They simply do not know their address, and they can't move into the tissue to kill any tumours. The second option here is that what tumours can very cleverly do is turn off these address signals. This is quite rare, but if this does happen, it means that these cells can no longer get into this tumour, and both the yellow and the purple cell get effectively lost, and this will once again let your tumour grow and spread. The other thing they can do is they can actually change their address so that your cells can no longer find their way to that tissue, and once again your cells get lost, meaning they can't make it into these tumours, and your tumours can then grow and expand. Finally, maybe your cell does know how to get to this party. But you get there and the door's locked. So you've not actually been invited to this party and you're not allowed inside. And tumours can do this to your immune cells by turning them off so they can't get in. So either you know how to get to the party and you can get there and do your job. You don't know how to get to the party and you're stuck outside. The party's then moved somewhere else and you go to the party and can do nothing about it, or you do know how to get to the party and you get locked outside. And these are different ways that tumours can manipulate the chemokines to help our cells get lost and help our tumours grow. But tumours themselves are also really good at manipulating these address codes to help themselves. So cancers will often produce a chemokine called CCL2. CCL2 is really good at attracting a cell type called a macrophage. Macrophages are normally really good, as macrophage stands for big eater, and what they normally do is they will come into a site of inflammation and they will eat up any bugs we see or any debris from cells that have died and clear it away to help keep us healthy. But cancers can actually manipulate macrophages into helping them. So cancers can actually trick macrophages into helping tumours spread to other parts of our body, which is a process known as metastasis. When this happens, tumours start to spread elsewhere, and that's what kills the majority of cancer patients. The other thing that cancer cells can do is they can actually turn on the receptors that allow them to move towards these different address codes that our body already has naturally throughout. When this happens, cancer cells can move to these locations and they can grow and once again undergo metastasis, which we don't want. But it's not all bad news. What if we could get the good guys to respond to CCL2? If we could, this would let the good guys always move into the tumours and kill them. But what are the good guys? Who are the good cells that protect us from cancer? And these are natural killer cells, which we'll call cancer-killing cells. So NK cells, or the cancer-killing cells, are designed in a way that they can tell if any of our body's cells are no longer healthy. So an example here, you have a cell that gets infected by a virus, and the NK cell can tell that this cell is no longer healthy, and that will kill the infected cell and clear it away. And they do the same thing with cancer. So NK cells function much like we might judge, say, a piece of fruit. So here you have a healthy apple and a rotten apple. You're always going to eat the healthy apple and ignore the rotten one, whereas NK cells function on the opposite, where they will eat up any rotten apples and remove them from the body, leaving only healthy apples behind to make sure we stay healthy. So how can we use this? So we can take some cells from blood that are in quite early development, and we can turn them back into a type of stem cell that can become any immune cell we want it to. We can then modify these cells so they always express CCR2, which is the receptor that lets cells move towards sources of CCL2, which is what is produced by tumours. This should let the good guys always move into the tumours and eliminate them before they become a threat. So what have we learned tonight? Well, our body is designed much like a fortress. It has walls to protect the inside, in the body's case that is the skin. And there's lots of foot soldiers, the immune cells, inside to help keep us healthy. But sometimes they can get a bit lost. So the research that I do is looking at ways to help the good immune cells get into tumours and kill them. And this should hopefully leave our body full of healthy apples, taking away all the rotten apples, which will then leave us nice and healthy as opposed to the opposite. So thank you very much, guys, for listening. I hope you enjoyed the talk, and if you have any questions, please put them down in the comments, and as George said, we'll answer them later in the show. So now I'll pass you back over to George, who'll take you on through the rest of tonight's show.
Thank you so much, Alan. That was great. But it's so strange. I didn't realize that cancers were so almost devious in being able to shut off the immune system and how it can change cells and how to migrate. But it's also great to see that we can use that to advantage, where certain cells, like say NK cells, are able to kind of almost bypass that. I think it was amazingly fascinating. Thanks for that, Alan. That was great. So in the intermission, we've had a few comments about some fun facts. I put one up earlier. I think especially in a time such as now, where vaccines are very much in the forefront of conversation, I think that the fact that the majority of a single vaccine is actually mainly water, I think is so fascinating. There's obviously lots of other bits and pieces that come into it, but much like a human, we're actually pretty much a very much majority of water as well, which I think is also great. I love Laura has also put in one, which is not technically a factually correct one, but I guess I can also see that when kids are in daycare, I can also imagine that a stress response or a non-stress response could definitely shift how your immune system may work there for sure. I also find it really cool that like certain external factors like stress or the circadian rhythms that you have during like different sleep patterns can really have an impact about how your immune system works. Another one whilst I'm here is that I forget where it's from, I think it's the BSI vaccine group, is that if you have a vaccine at certain times of the day, whether that's in the morning, afternoon, or the evening, which works into your circadian rhythm, it'll actually change how you react to the vaccine based on actually the time that you had it taken, which I think is really fascinating. There's some really cool data out there on that, which I think is cool. I also, Paul Doherty is wondering how many pints can impact your immune system. Well, I guess when you think about it, alcohol as a generic drug is a depressant. And I can imagine in certain instances, I don't know if you could do it for a human study where you have people who have drunk X amount of alcohol at different quantities, say I'm about a half a pint in. I wonder if my immune system is either slightly more immunosuppressed based on the general depressant factor of alcohol, or if there's a threshold point to see if that has anything to do with it. I think that would be really cool to do. It'd be a long study though. I can imagine you get a lot of willing participants considering pubs being reopened, at least outside for drinking currently in Scotland here. So I think that would be great. That'd be great fun. Just having a look to see if we've got any more come through. Not that I can see at the meantime, but thank you for putting in what you've put so far for our little immunology fun fact section. Again, if you've got any questions for Alan's talk, please feel free to put them into, so Paul's happy to volunteer, love that. Please feel free for any questions for Alan's talk, please put them in the chat, and we'll have those for our Q&A session at the very end with both Alan and Marieke. And with that, I will hand you over to our second speaker of the evening, to Dr. Marieke Pingen, who's going to talk to you about mosquitoes and the pesky viruses that they transmit.
Thanks very much, George, for that introduction and for those great fun facts. Also, thank you, Alan, for your really interesting talk about how the cells of our immune system move. In my talk about mosquito-transmitted virus infection, we'll see how these moving cells and other parts of our immune system can maybe help us stop mosquito-transmitted viruses at the bite site. Did you know that mosquitoes are indirectly the deadliest animals on the planet? They even surpass humans. This is because mosquitoes transmit infectious diseases. Malaria, which is a parasite, is transmitted by mosquitoes to over 200 million humans a year. and over 400 million people get virus infections every year transmitted by mosquitoes. As I'm a virologist, I will focus on these virus infections today. Scarily, these mosquitoes are expanding their territory. This is because we increasingly travel the world, maybe not last year, but in general, and also because of transport of goods. And this way, mosquitoes, and especially their eggs, which are very resilient, are being sprinkled all over the globe. And if they land somewhere where they can survive, which is an increasingly large area due to climate change, they can start new colonies and continue living in expanded territories. And with these mosquitoes also come the virus infections. To make matters even more complicated, this is not just one virus. There's actually over 100 different viruses that can be transmitted by mosquitoes that infect humans. Just one of the recent examples is the Zika virus, which you may remember from 2015, when a large outbreak resulted in thousands of babies being born with microencephaly because their mothers got pregnant during the infection. Also, every year, hundreds of millions of people get dengue virus, which can cause a severe hemorrhagic fever. Whilst many of these infections are relatively mild, a lot of them can cause very severe temporal arthritis, infection of the brain, or other very serious complications. It's important to realize that all these these over 100 viruses are very dissimilar to the immune system, as illustrated in these graphs. The viruses all look very different to our immune system, and they also replicate in different ways, which makes it very difficult for us to develop drugs and vaccines against them. And as a result, there is currently no antiviral drugs and only a handful of vaccines available for viruses that are transmitted by mosquitoes. It is very difficult to prepare for these outbreaks because we currently can't predict which virus will be the next big one to cause an outbreak. And as you can imagine, it's impossible to prepare drugs and/or vaccines for 100 different viruses just in case. So traditionally, drugs and vaccines are generated specifically for single types of virus. And we would like to try a different approach. So we think that by understanding our immune response to mosquito bites, gives us tools to activate our skin defense mechanism at the mosquito bite, and therefore by helping our skin immune defense, we can fight multiple viruses at the same time instead of spending lots of time and lots of money at lots of different drugs for any single virus. So by looking at the common denominator, the mosquito bite, we hope to fight these viruses. For that, we first need to understand what happens in our skin when we get bitten by a mosquito. To help me explain this, I have this beautiful cartoon mosquito, which is now biting our graphical skin. So what happens in our skin? It's actually only the female mosquitoes that bite us, and this is because they need a blood meal before they lay eggs. They use their bitey bit or their proboscis to pierce our skin repeatedly. So as Alan explained it, they go through our thick skin wall that protects us against infections. And by this repeated piercing of the skin, they're looking for a nice juicy vessel so they can drink our blood. We call this probing. During this probing, the mosquitoes deposit saliva. Saliva is a very complex cocktail with over 1,000 ingredients, many of which are in the saliva to help the mosquito from feeding, for instance, by preventing our blood from clotting too quickly. Recently in our lab, we found that one of these ingredients makes our vessels go leaky. That's indicated in this graphic with the leaky blood vessel. This leakiness also causes a bit of edema, which means that there's some water retained in the skin, which we call edema, and we get a classic mosquito bite, which you probably recognize. We wanted to further investigate what is actually happening in the skin at the time that this mosquito bites and deposits their saliva in the blue dots and the virus in the green dots. To do so, we put mosquito-bitten skin under the microscope. This is a piece of skin, and on the right hand and the left hand side, it is healthy, but here we have a mosquito bite. We recognize the mosquito bite by this red clot, which is red blood cells. So for instance, if you have a scab, that's made of red blood cells that clot and prevent all your blood from leaking out. As you can see here, there's a streak of purple dots. And when we zoom in on those, we recognize these cells as neutrophils because they have a very specific type of nucleus. So what do these neutrophils do? They are the first line of defense. So they are very important cells that help with any sort of damage control in the skin, but also in other areas of our body. So anywhere where there's damage, the neutrophils come in really quickly to help organize the cleanup of the damage that was made by the breach of our fortress. Furthermore, they call for help from other immune cells. So after determining what type of damage they need to deal with, they send out the right molecules for recruiting the right type of cells to deal with the mess that they found. These neutrophils also contribute to the edema, and this causes the virus to be retained in the skin. So what cells do the neutrophils call for? The next ones on the scene of the crime are the macrophages. As Alan described them, big eaters. They are at the second line of defense, and they're very important in protecting the skin against infection. They literally eat anything they don't like, so they can then present it to the rest of the immune system and explain what's going wrong by, at the same time, making it safe by eating it. So when we infected skin with a virus that makes a green protein and stained for macrophages, we found something very interesting. Here we see blue dots, which means the cell nucleus. The red shapes are the macrophages. So as you can see, they're very irregular, and that's because they are always on the hunt for things to be eating. And as you can see in green, which are the virus-infected cells, there's a lot of overlap of the red and the green color. And this means that actually, whilst the macrophages are there to protect us, they are infected with the virus and they are making more virus. So unwittingly, whilst they're there to protect us, the macrophages get infected. So to summarize what happens in our skin when we get bitten by a mosquito, after the saliva is deposited, neutrophils and macrophages arrive in the skin to protect us and to repair the wound damage of the biting and the saliva. The virus then replicates in the skin, which is amplified by the presence of macrophages where the virus can replicate in. From the skin, the virus spreads to the rest of our body. And sadly, the skin antiviral response seems to be too late because the virus is already spread to the rest of our body. But does this actually mean that that we get more sick? To answer this question, we infected mice, and I'll explain how this graph works. So on the y-axis, we see the percentage of mice that remain healthy. So 100 means 100% of the mice are still happy and healthy. On the x-axis, we follow the infection. So we check our mice multiple times a day to see if they show any signs of being ill. In the gray line, we see the virus alone. So if we just inject the virus, nothing happens. All the mice remain perfectly happy and healthy. However, if we inject the exact same amount of virus in the same location of the mouse, but in a mosquito bite, then we see that about half of the mice show clinical signs of disease, so they're not well. And that shows us that the mosquito bite is really important, not only in replicating virus in the skin, but also in spreading the virus through the body and causing disease. So you would maybe wonder, why does the skin not do anything to fight back? We found, when we looked at this in detail, that if you look at the amount of virus, it very rapidly peaks, as shown in this green line, which shows the amount of virus in the skin. And by the time the antiviral response in this pink line actually only comes up later when the virus is already spreading through the body. So we were wondering if we could give the antiviral response a bit of a boost and therefore maybe decrease the amount of virus in the skin, and hopefully that will help prevent the virus from moving from the skin into the rest of the body and cause disease. To investigate this, we used Aldara. Aldara is a already approved cream that is frequently used for people with genital warts, which are caused by another virus. So basically, you can topically apply this because it's a cream, and generally it's very well tolerated with only mild side effects. To investigate whether this cream would work in this context and whether we can boost the immune response and help prevent disease, we used our mouse model. So mice were bitten with mosquitoes and then immediately injected with virus. And 1 hour later, we applied this cream on the skin. We then followed the mice and how they were doing and how the virus was replicating. The first thing we did was measure the amount of virus in the skin. For this, we used PCR tests, which is the same as what is used to test for coronavirus infection. And it allows us to really precisely measure the amount of virus in the skin. So on the y-axis here, you can see the amount of virus genomes that we found. And here in black, we see the amount of virus without treatment. Whilst here in green, we see that the mice that were treated with the cream are doing much better and have much less virus in the skin. Of course, it's also important to know whether the virus is then going into less virus ends up in the blood. So we looked at that, and indeed, there is much less virus in the blood of the mice that were treated with the cream. We then used the nastiest variant of this virus that we could find, and we looked whether this cream could protect the mice. So this graph works just as I've shown you before. So on this axis, we see the healthy mice as this means 100% healthy mice, and then we follow them over time. And as you can see in the black line, the mice with just virus and no treatments became sick quite quickly. whilst the mice that were treated with the cream in the green line were much more protected against virus infection. To investigate why these mice became sick, we looked at their brains. And as you can see here on the left, this is the brain of a mouse that was infected with a virus that makes red proteins. So all these red dots in the mouse brain are all areas where the virus is highly replicating, explaining that these mice were becoming unwell. On the right-hand side, we see the brain of a mouse that was treated with the cream. And as you can see, there's no virus, so it makes sense that the mouse was feeling fine. Obviously, a mouse is not a human, and whilst we use mice as a model because a large part of their immune response is very similar to ours, they're just not the same. Therefore, we use the human skin model. We obtained healthy skin biopsies which were taken by a dermatologist. And these skin biopsies are basically small circles of skin, and these pieces were cut in half, where both halves were infected with the same amount of virus. 1 hour later, just like with our mice, we applied the cream on half of the skin, and then both halves were allowed to, were continued to be cultured in the lab, and we tested the skin for how much virus was made. For these experiments, we used 2 human pathogens, so 2 viruses that have caused outbreaks in the last 2 decades, so Zika virus and chikungunya virus. And as you can see in the schematics, these viruses are quite different, and they're from different virus families. So it's very important for us to test whether our treatment worked on different families of viruses. In these graphs, you can see the amount of virus in the y-axis, and each dot means 1/2 of a skin. So in this case, the 1/2 of the skin made a lot of virus without cream, but with cream, it made very little virus. And we saw this for all the pieces of skin where we applied treatment, there was much less virus in the human skin. This shows us that this is a promising way of treating human skin as well. So to summarize all of that, what happens in our skin when we get bitten? First of all, there is an immune response that focuses on wound healing rather than antiviral response. And unwittingly, this benefits the virus in spreading through our body. So can we boost the skin in helping it to fight viruses? Yes, this actually looks really good. Activating the immune system at the bite site can decrease different types of virus infection in both mice and in human skin. An important disclaimer here is that this hasn't been tested in humans, and it would take a lot of effort and work still to make this into a real therapy. But we think that this does demonstrate that fighting the mosquito bite is a really good approach of fighting many mosquito-transmitted viruses with a single drug, which would greatly benefit the treatment of all these viruses. Thanks very much for listening. Also, thanks for all the collaborators and teammates that have been involved in these projects. And back to you, George.
Thanks, Marieke. That was fascinating. I think it's so great that, like you say, you can have something that is applied to the skin that actually helps reduce viral spread, the virus that actually goes to the brain. So if you treat the actual site that it happens at, there's some kinetics that then mean that as it's then trying to travel maybe up to the brain, it's less able to do that. I think it's fascinating. I think that was really, really cool. Thank you so much. So now this brings us to the last segment of our show, which is our live Q&A with both Alan and Marieke. And during both Alan and Marieke's talks, We've had some lovely questions come through from the public, which is amazing. I thought I would start things off. So I have a question to Alan, which is, how do tumours actually turn off immune cells?
So that's really an interesting question there, George. So if you think of your immune response, if you get an infection, you want to get your cells there as quickly as possible to kill the threat. But you don't want that immune response to go on forever, because that would just be detrimental. Because as you know, if you get an infection, the areas kind of get really red and inflamed, for example, and it's sore to touch. It's not the most comfortable thing. Or if you've got the flu, you've got fever, you don't want those things going on forever. So our body has mechanisms where it can turn on certain proteins that actually turn off immune cells themselves. Yeah. So that sort of thing is called an anti-inflammatory. So our body can make anti-inflammatory proteins that then dampen down the immune response, so you get a peak of cells activating that then comes back down and your body returns to normal. So cancer cells can actually produce these proteins so that as cells infiltrate into the tumour, they're met with these proteins that tell them everything here is okay, you don't have to do anything, basically go back to sleep. So cancers are good at doing that by producing these molecules that our body would use naturally to do the same thing once we've cleared an infection.
That's so cool. Again, it's another kind of world where you just think about what actual cells can do in terms of just general communication of how to turn things on and off, and how sometimes that's so easy, but at certain times it's so intricate as well, of how certain mechanisms have to really be playing along with the whole grand, almost like an orchestra kind of thing. Even if one person's playing out of key, everyone will notice. So it's kind of that being like, you have to stop that person to get the rest of everything coming back together again. That's so cool. I love that. I think looking down the list of questions, we have a question from Ailish McCafferty, which is also a question to Alan, and she's asking, are there any therapeutics currently in use which work by targeting either chemokines or their receptors? I think that's a really good question.
So the short answer is yes, but there's not any in the way that we are trying to do it, where we're trying to modify how cells themselves can see chemokines by giving them extra receptors. But the most common example you hear of is a drug called Maraviroc that's designed to treat HIV. So this is going to be a big tangent here, but HIV infects cells of the immune system, and HIV infects them by using chemokine receptors to get inside your cells. So what Maraviroc does is it binds to CCR5, so it blocks the receptor. And this then means the virus itself can't bind to it and can't infect your cells. So that's one of the treatments you can use for HIV to stop the virus being able to infect more of your cells. The problem with blocking chemokines is, you saw that diagram I put up during my talk where the chemokines bind to multiple receptors and multiple receptors interact with multiple chemokines. So because there's so much cross interaction, you can end up with off-target effects. Or if you block one that you think might make a huge difference, what you actually find is that one of the other chemokines can almost fill that role instead. So there's a little bit of what we call redundancy, where sometimes the receptor that does a job, there might be another receptor that can then do that same job that we've not realized. So they're actually really tricky to interfere with in that way, which is why we are trying to take it in the way of, well, what if we give a cell an extra receptor that will let it go to a certain source, rather than can we stop these receptors from working, which is the kind of general way you would take a therapeutic like that.
Yeah, I mean, both you and Marieke both know how difficult it is trying to target chemokine receptors from being in CRG. I don't envy how difficult that must be. But yeah, when you showed that diagram, it generally does hurt my head when you see about how many different chemokines can bind to X amount of different chemokine receptors, and they're being like, but then there's like 5 different ligands for one receptor, then what happens then? Because if you get rid of one, you've got another 4 that can do the same job. So why does that then matter? And yeah, do you sequester a ligand? Do you block a receptor? Do you move one around? It's a whole different kettle of fish when it gets onto that. But yeah, I think, like you say, doing it that way, I think is great. Oh, Laura from Pint of Science had a question, which I think is also really interesting, is why do mosquitoes in one way bite me and not my husband when we're together? Which I think does raise an interesting sideways section of, actually, are there any differences between, say, male and female, or other things that causes mosquitoes to bite more than something else?
There's actually quite a lot of factors, but it basically boils down to how mosquito thinks that you smell. So there's certain molecules that you might make that mosquitoes really love, or maybe there's some molecules that your husband makes and that they really, really despise smelling. So from this limited information, I can't say whether they're attracted to you or repulsed by your husband. But basically, mosquitoes are very picky when it comes to smell.
I wish that when I go camping, if I knew that mosquitoes didn't like someone being smelly, I would just not bring any deodorant so that I would never get bitten. That would be the ideal.
So sadly, it's not how clean or how smelly or how stinky we are, but mosquitoes can pick up really small molecules that are in our scents that we're not aware of. But they really home into it, or they get really disgusted by it.
I just can't deal. We've all been there. Not for that specific thing, but in certain aspects. I think Paul Doherty's had a couple of questions too, which is great. One to both of you is that he's been thinking about viruses, and he's been asking can viruses be used to treat cancer or to curb infections from other viruses? So that's the floor for both of you.
Ooh, do you want to take this, Marieke, or shall I?
Do you want to take it from a cancer angle, and I'll talk about viruses?
I can certainly try. So, in a way, potentially, but one of the problems with the immune system is that the part that I showed you that there's an innate immune and an adaptive immune system, and generally your adaptive immune system will do a lot of the specific work for taking out specific viruses or cancers. So if you use a virus, you'll make an immune response against that specific virus to kill it, which won't necessarily help you deal with a cancer. But there are possibilities where you could maybe modify viruses and stuff in such a way that they're not going to threaten somebody's life and that they're easily deal withable, but that they have proteins that might be expressed by a cancer cell or another virus or bacteria that will then help your body make an immune response against the cancer cell by using the virus as like a kind of vector or a fake cancer cell that your immune response will make will fight against that specific protein, and it should then translate onto the cells. As with all things biology, it's never really that simple because there's lots of other bits that come up that can get in the way. But in theory, yes.
There is actually lots of research on this to use vaccines to use viruses as sort of a vaccine and against people's cancer. So what scientists are trying is to chop up someone's cancer and then make a personalized, this is what your cancer looked like and you should be angry with it, immune system vaccine response. As far as I'm aware, this is not done for infections from other viruses. I've never heard or read anyone trying that, but it's an interesting idea. What we do is use vectors based on viruses. So a vector is a circle of DNA, and we use a lot of viruses that are not dangerous to make vaccines. So indirectly, yes, you can.
I think to add to Marieka's point, so I work on flu as a model for my work in viruses, and there's a very cool paper that's come out from Pablo Murcia, who is at the Centre for Virus Research in the University of Glasgow, and they look at co-infection. So they look at different viruses and how that changes the fitness of a certain infection. And the paper they put out recently this year in an in vitro model, so looking at cells cultured in a dish, they show that if you infect with rhinovirus, so that is the virus that causes the common cold, if you infect with rhinovirus first, and then you were to infect with coronavirus, one of the same strains that's happened worldwide so far, it actually discourages the replication of the coronavirus if the rhinovirus is there first. So there's this whole competition of resources that happens between viruses I think is really fascinating. And that's the absolute epitome of my knowledge. I don't know a lot about it either, but I think it's a really cool thing to go through. Very, very cool thing indeed. And we've got another question from H. Davis. This is for Alan. And he asks, with the increasing number of receptors on the cells, are there any possible off-target effects? For example, an autoimmune response that you could get from this?
So that's always a risk with this kind of thing, that you might have off-target effects, which is why anything that we would develop such as this would go through rigorous testing to make sure there weren't any off-target effects. But to bring up your specific example of autoimmunity, autoimmunity is a tricky one because it's developed against an antigen that your body expresses naturally. So, for example, in rheumatoid arthritis, it will attack the bones. So your body, to form an immune response against that, would have to see that protein in a bad light. So it would have to see it in kind of an inflammatory situation. So normally our body's immune cells are very good at knowing what is actually our own cells and won't attack them. If they get these kind of mixed signals where they mistake it for a virus, they can. So in that regard, there might not be many off-target effects because we're not actually inducing inflammation ourselves. It also depends really what cells you use. So we're using these natural killer cells, or the ones that can kill cancer, and they won't function in the same way to make a full-blown autoimmune response, but there could be some off-target effects if they kill the wrong cell or whatever, which in theory shouldn't happen. The other thing is that the receptor that we plan to use, or that we do use, CCR2, it really only interacts with CCL2. And CCL2 is what we call an inflammatory chemokine. So you get different types of chemokine. We get inflammatory ones and ones that are called homeostatic. And the homeostatic ones are always expressed to let cells go about like these address codes. And the inflammatory ones are only really expressed at points of inflammation. So if you have an infection going, they're used to bring cells in. So they modify the address code to let cells know, oh, there's danger here. So by targeting ones like that, it should hopefully reduce off-target effects because your cells should only go to sites where they're actually needed to do a job at that time, if that makes sense. So hopefully it does.
I guess, like you say, like there is some redundancy in the chemokine receptors, but also there are other examples of where there are specific ones where it's just one ligand, one receptor. I was looking at ones of like CCL9 that only binds to CCR1, and like that seems in the chemokine world very simple, which usually is not.
Yeah.
What else we got? So we've got a question from Alice Burrows for Marieke on a kind of human versus mouse model point of view. So she asked, if you can test on human skin, which would surely be more accurate than testing on mice and more ethical, why do we test on mice?
That's a great question. And one of those reasons is actually all the things that were explained by Alan. Whilst in the skin, we have the cells that are in the skin, we can't ever investigate which cells are being recruited to the skin if we don't have the rest of an organism attached. So if we only look at a piece of skin, we can never fully comprehend what the response will be, because the skin might scream like, give me macrophages, give me neutrophils, give me NK cells, I want anything. But they can't go there because they're not in the model. So whilst human skin definitely has advantages, including that ethically it is preferable, and we do anything we can to reduce and limit the number of mice that we use. But for certain things, especially when it comes to something complicated as cell migration, we sometimes do need a full organism to fully understand what's going on.
Like I say, they both complement each other in different ways, and that's all how for it to work. Linked to that, I guess you've got Ailish with another question for you, which is, could what you've learned about the response to these virus infections maybe instruct the design of a broadly effective vaccine that works against many mosquito-borne vaccines? I think that's a great question.
That is a great question. And actually, that is something that we're really interested in. Due to time and especially due to time limitations, I couldn't go into the saliva story a bit more, but we had a very good PhD student called Daniela Lefteri, and she did all this work on the mosquito saliva. And one of the interesting things about it is, like, if we can block the molecule that makes our vessels go leaky, and that almost single-handedly causes this bite enhancement of mosquito infection, if we can block that with a vaccine, could we then block that the immune response helps the virus? We have not yet fully investigated this, but if you know someone with a pile of money, I'm sure we can make things happen.
It's like the never-ending to-do list of experiments that I wanted to do that would be amazing, which would work. Kirsten Bloomer has asked for you, Marieke, the data that you generated in mice and from the skin biopsies look very promising so far, which is great. Do you have any concerns regarding its efficacy in human trials?
So human trials aren't exactly my my forte. What we do know is that this specific cream is very well tolerable, so people can apply it to larger areas repeatedly without getting really nasty side effects. It's important to also mention that this, for us, is a proof of principle. So rather than buying shares of this specific drug, we'd rather see this as a step forward and a different way of approaching fighting mosquito-transmitted viruses.
Like, you don't know, like you talk about translational models, etc., about how things are going to go. There's only so much you can tell from a mouse model to then translate into humans, and there's always going to be a bit of a risk in terms of that translational side of things. But I guess if you can nail down what's actually happening in a mouse model, that's the best thing you can go towards going into a human model. I think
exactly. Yeah. So that's what we focused on. We focused on what we do well, and we're hoping that people who are good at clinical trials might get inspired and take it from here.
Here's to good science collaboration. I think that's great. So I'm looking at the time, guys, and we're almost done. But thank you so much to both Alan and Marieke for both your wonderful talks. And thank everyone for all of the questions that you put through in the chat. Sadly, we actually didn't have the time to go through them all this evening. However, you know, Marieke's Twitter and Alan's Twitter are both on the page. If you want to get in touch with them, that's where to go. Put your questions to them. That would be great. And yeah, just before we head off this evening, guys, one thing I'll happily do as a shameless plug for the University of Glasgow is we actually are hosting another session tomorrow night for the Beautiful Minds theme. And that is called What Plagues the Mind, Infections and Malfunctions. And that's where you're going to have two very talented PhD students, both Lorna Hayden and Miriam Scarpa. And they'll both be talking about interesting findings from multiple sclerosis studies and the creation of a roadmap for better dementia research, which I think is going to be really interesting. And I'm very excited to tune in tomorrow at 7, which will be great. But in the meantime, guys, I've been your host, George Finney. We've been Pint of Science for Our Body from the University of Glasgow. And I just want to thank everyone for tuning in tonight. And here's to more great Pint of Science shows for the future. Thanks very much. We'll see you soon.